All right, folks, now that you already understand to some extent how genes are regulated in eukaryotes like ourselves, it's time to talk about bacteria. Now, it might have been easier to start with bacteria, but now you're going to find this is going to be way simpler. In our cells, with 23 pairs of chromosomes, 6 billion base pairs, there's so much complexity. In a bacterium, this will be simpler to look at. It really is. And in many ways more efficient. There's just one chromosome, which you already know. In addition, there might be additional little plasmid loops of DNA. But instead of there being billions of base pairs, we're down to millions. Instead of there being, say, 20,000 protein coding genes, we're in just a handful of thousands of genes. So there's a lot less to look at or control here. So in a bacterial cell, we're going to have on the right our single looped chromosome. And sorry that the colors don't match what's on the left. We are going to have promoters. That's what the green arrows represent here. These are promoters where RNA polymerase will begin transcription. But this is where we begin with it being way simpler than eukaryotes. There are generally no histones. We don't have to worry about DNA condensation. Even better, you don't even have to worry about transcription factors being necessary for RNA polymerase to bind to the promoter. RNA polymerase in bacterial cells typically can bind directly to promoters, no transcription factors necessary. Now, if the RNA polymerase binds at some basal rate, it will do transcription. Now, what you're going to see is going to be a little bit different than eukaryotes from here on out. The sequences that come after the promoter, you'd expect a single gene. Here it's a little bit different. There's the promoter. There's a sequence that's either part of the promoter or overlapping and going after that called an operator. We'll get to that later. And instead of there being just one promoter and one gene, each promoter in a bacterial cell often has more than one gene after it. And you're wondering why this makes any sense. Honestly, it makes more sense than what eukaryotes have in many ways, in that what we're going to do is we're just going to turn on a bunch of genes or turn off a bunch of genes all at the same time. There's an efficiency bonus to this. Now, this whole cluster is called an operon, and that's a word you do need to know. It's an ancient term. Back from the 1950s. And it's a cluster of genes all controlled by a single promoter. Now, the efficiency bonus is oftentimes there are multiple enzymes needed for any process. So instead of turning on 5 separate genes for 5 separate proteins using 5 different promoters, why not just put them all together? Now, an example that I'm going to make up here in just a moment deals with DNA synthesis. And again, I'm making this up. This isn't actually accurate to reality in E. coli. But you've already learned about DNA replication. And you know that for DNA replication, you need a helicase, and you're going to need a topoisomerase, and a primase, and a DNA polymerase, and another DNA polymerase, and a ligase, etc., etc. There are so many proteins you need for DNA replication. Well, if you are going to do DNA replication, why don't you just turn on all of those genes together? So that's what we're going to have here. We have a single promoter, a single operator, which we'll talk about later, and then the helicase gene, the primase gene, the DNA polymerase gene, and the ligase gene all in one operon, one sort of cluster that we can turn on together. Now, what's going to happen is by the time the RNA polymerase gets to the end, it's going to start at the promoter, as you'd expect. When it gets to the end, it's going to have made one long mRNA transcript. No capping, no tailing, no splicing required. And what's really cool about this is because we're talking about a bacterial cell, no nuclear envelope or anything of that nature, the ribosomes are right there. They can immediately bind to the mRNA as it's being transcribed. So transcription and translation are happening simultaneously. In fact, on any mRNA, as it's being transcribed, as the RNA polymerase is making that yellow mRNA, ribosomes are already on it making protein. Now, this is where my animation starts to get really ugly because I'm at the limit of what my stylus can do in this particular software. But these green things are supposed to be the new proteins coming out of the ribosomes. Sorry for it being so ugly. But again, what we're going to do is we're going to have a cluster of genes, all for the same process, in this case DNA synthesis. We're going to turn them all on at the same time by putting an RNA polymerase at that promoter. The RNA polymerase is going to transcribe all the genes onto a single RNA, single mRNA. And as it's doing so, we're going to make all 4, 5, 6 of those proteins simultaneously using ribosomes. That's the general idea here. Okay. Now, this particular operon, DNA synthesis, DNA replication that I'm making up here, you probably want it on all the time because bacteria reproduce so rapidly, as rapidly as, every, half hour or an hour in some cases, depending on how much nutrition there is available. Other operons, other groups of genes, you don't need to use all the time. Again, certain sets of genes like DNA synthesis, you want on all the time. Other ones, you're going to use less often. So up at the top, I just showed you what an operator is for. An operator is an annoying vocab term, and all it means is repressor binding site. So up at the top, that top operon, you see sort of a triangular shaped protein that bound to that DNA sequence. That's your repressor. And so all it's doing is it's binding right in front of the promoter or at the end of the promoter or in the promoter even. And it is very simply a physical roadblock. Even if the RNA polymerase binds to the promoter, it cannot get to the genes. It is physically blocked from binding or transcribing through the genes. So all of those genes, the entire operon is off all at the same time. Not one gene, not two genes, all of them in that operon. The efficiency is, again, there's no point in making one enzyme for a process if you aren't going to make the other ones. So if you don't need that system, just turn the whole thing off. The sort of canonical example of this is something called the lac operon. It's not the most easy thing to understand, but you're going to see it in various online resources. It was the second one of these operons that was identified. This is the one that produces the enzymes to help bacteria digest lactose. So that's milk sugar. So if you don't know that, in milk there are sugars, and the primary sugar is lactose. And most E. coli bacteria, honestly, don't have a lot of lactose as a food source. E. coli living in your gut doesn't have access to lactose because most of you aren't drinking milk all the time. Adult humans don't, historically or evolutionarily, adult humans don't drink milk until the last few thousand years. So in E. coli bacteria, which is a gut bacteria in you, these genes don't need to be on. Bacteria aren't using lactose and don't need the digestive enzymes for breaking down milk sugar. So this particular set of genes for lactose digesting proteins is typically off. The repressor is bound in front of the promoter at that operator sequence, and all these genes are off. In comparison, there are other operons, other sets of genes that are almost always on. And that DNA synthesis or DNA replication made-up example is one that's like that. The example you'll see for operons that are typically on is going to be called the trip operon. There are lots of these. This is just the first operon that was identified in Paris in the 1950s. And so this is the set of genes used to produce an amino acid called tryptophan. And this is typically on because E. coli has a hard time getting this particular nutrient. So it's always using these genes. But if they run into a particularly massive food source with a bunch of protein in it, lucky E. coli, they can use the tryptophan to shut down that particular operon and save themselves all of the hassle of making those enzymes. But we're going to move on from now because I want to keep this video under 10 minutes. If you want to read the text, feel free to. but I'll answer any questions in class next time. Best of luck, everybody. See you soon.